The link for the attendance. Please mark that. Okay. So in today's session, I'm going to talk about the NCBI Gen Bank database. So do you have any idea about this NCBI database? Or somebody can tell me what is the database? Please unmute yourself. What is the database according to you? So, yeah, tell me. So, database is very important. Why? Because this databases is storing the information of each and everything, okay, whether it is related to genomics or proteomics, literature, right? So, again, like if I ask you to design a primer, so for that, what you require? You require a sequence. And that sequence, if it is not present in the database, you cannot design the primer. So, database is a center of research, and in the database, first database we are going to see that is the NCBI, which is one of the primary database. Why primary database? Because anyone can submit the data here in the NCBI. So this NCBI stand for National Center for Biotechnology Information, which is maintained by the National Library of Medicine. So you can see there is a drop down menu here. So these are some databases that is present here in the NCBI. Again, if you want to get the mutation information, that time you can go with the SNP database. If you want to download any data, you can go with the SRA. If you want to get a protein information, you go with the protein, then genome, gene, DB So these are some databases that is already there in the NCBI. So you can see on the left hand side again. So this type of information you can get related to the medicine, homologies, literature, protein, sequence analysis, and also if you want to get any training videos also, you can get that also. So out of all these resources, you can see on the right hand side, these are some popular resources. Why popular resources? Because these resources are highly searched by the user. That is why out of all these resources, these are only some popular resources. That means most of the researchers or maybe any user, they are searching information related to these, okay, most of the time. In the center of this tab, you can see there are 6 functions. First function is the submit. Submit means, as you know, like NCBI is a primary source. That means it is an open source and anyone can submit their data. Then download. As NCBI is containing lot of data in it and we need to download, right? So mostly the data is free of cost, and whatever the data you want to download, we can download it from NCBI. Then learn. So NCBI is creating a variety of education such as webinar, seminar, or there can be a tutorial videos also. So which are totally free of cost. So if anyone want to learn anything, go with the learn function of this NCBI. Then next is it develop. So NCBI has developed many tools. So it is having its own API. You can see it stands for application program interface, which is helping the user to create any application or the tool. So next is the analyze. So as the name suggests, its purpose is to analyze, as it is a collection of data. If anyone want to analyze the data, NCBI will come with the wide variety of analyzing the data tool, which will allow the user to manipulate and analyze the data. The next is the research. So if anyone want to research with the NCBI, you just have to submit your research paper or any idea you can share. And if they found interesting, they will help you in the publication also. So let's see how to retrieve the information from the NCBI. So I hope everyone has opened this page, NCBI. Yeah. So our first exercise is to search for the gene PRNP. So gene we have to find out PRNP for Homo sapien. So Homo sapien is a scientific word for the human. Now in the drop down menu, you have to select the gene from here and type PRNP. Mm, is it opening in your system? Is it causing same error in your system as well? Can you try in your system? So our question is to search gene PRNP for Homo sapien. Please mute yourself. Now you can see after the drop down menu, you have to change to gene and you have to write PRNP and then search it. Then total hits you will get for the PRNP below. And on this right hand side, you can see top organism here. So again, if you want to be specific for the particular organism, you can select, for example, Homo sapien or maybe the mouse, cow, rat. So it's again totally up to you which for which organism you wanted the entry. You can select that and you will get a results. But the most important is this table. You can see here in the first column it is written name and the gene ID. So name is what? The PRNP. And along with that, the gene ID you are getting. If you know the gene ID also, you can get the information for the PRNP. So what is the description? That is the prion protein. So why it is called as the prion protein? Because this PRNP gene is making a protein that we call it as the prion protein, that is PRP, which is active in the brain cell, and it also include the transport of copper into cell and protecting the brain cell from the injury. That is why they are calling it as the prion protein. So what is the role of the protein behind that? That is, it is playing an important role in the synapses, where synapses means what? There is a small gap between the nerve cell that allow them to communicate by passing the messengers or messages to each other cell. That is what the synapses. Then it is written Cano blood group. Why Cano blood group? So Cano is the like antigen. It is on the prion protein encoded by the PRNP gene. So location, this PRNP protein, if somebody asks where it is present on chromosome 20. Along with that, you are getting the NCBI chromosomal ID. This is the start position and the end position on that chromosome 20. A lysis is nothing. This is the alternate symbol or the name of the gene. Earlier, this PRNP protein, it is also called it by these names. MIM, it stands for Mendelian Inheritance in Man. So it is one of the database, MIM. So again, if you are interested with the information related to phenotype, that time you just go open the MIM and put this ID. You will get the all the phenotypic nature of this PRNP in this from the MIM database. So now our question is to search the PRNP for Homo sapien. And if you have noticed in this table, it is the entry for the human, Homo sapien, right? So we are interested with this in entry. You can open that. Now you can see the table of content is just the index page of this report. And this is the full report of this PRNP protein. Now again, this is the PRNP, prion protein, full description, the gene ID, and it is recently updated on 4th of Jan 2025. So official symbol, it was given by this HGNC committee that we call it as the Hugo Gene Nomenclature Committee. It is providing a unique name to the gene. Again, official full name, prion protein, it is given by the agency committee. So as you can see, agency one nomenclature committee. So it is assigning a unique gene or the description to your genes. Now primary source is nothing. So it will give you only the information related to this PRNP. Okay, firstly, when this PRNP was submitted in the NCBI, that first information you will get from this primary source. And then ensemble MIM aligned genome. So again, these are three database. Ensemble, in the ensemble, you will get the functional information of those PRNP. or maybe the regulatory information, or you can see the annotation at the genome, gene, protein level, including the genetic variation or homologous information you might get from this ensemble. But if you are interested with the entire genome of this, you can go with the Alliance Genome Database. So now further you can see gene type is the protein coding. So that means this gene is coding for the protein. That is why it is written protein coding here. RefSeq status is reviewed. Reviewed status mean that particular record has been re-verified or reviewed by the NCBI staff, and it has undergone additional functional review beyond the sequence review that validate all the records that has been submitted here in the NCBI. From the experimental level till the research part, each and everything it is cross verified by the expert panel which is present here in the NCBI. Then only it is coming under the reviews. They are having some valid proof and evidence with them. But if you see sometime unreviewed means it has not that much evidence with them. Still, the research is going on for that particular gene, and the validation is also going on for that particular gene. Incomplete information is there, or maybe you can say assumption is there for that particular gene if the unreviewed word is written. Next is the Homo sapien. So lineage is nothing, it is a taxonomy classification. It is grouping the biological organism based on the shared characteristics. They are ranked in the kingdom, phylum, class, order, family, genus, species. So also known as, these are the aliases, or you can say the alternate name for the PRNP. So previously this PRNP was known by this different name, and in different databases gene was identified by different name. So for example, this CGD was recognized here in the ensemble, GSS was resembled in the MIM, PRP is what resembled in the Alliance genome. So it's very difficult for the user to remember each and every name, right? That is why this agency came further and they have given a unique name to the protein. So nowadays we are not learning this aliases, or you can say the alternate symbol of the genes. So directly you can put this PRNP in any of the database, you will get that information. In the summary section, these are just telling you the protein coding, on which chromosome it is present, which type of disease they are causing. So those information you are getting here in the summary section. So expression, this RPKM is what read per kilobase of transcript per million reads mapped. So it is a unit of a gene expression. So they have like worked in the 24 tissues, and what they saw, the PRNP was showing higher expression here in the ovary. You can see the RPKM value is, this brain 148.7. So PRNP is showing higher expression in the brain than in the ovary than in the 24 tissues. So this is how we are searching any gene from the NCBI. Now, in the location. So can somebody tell me on which chromosome it is present, this PRNP? So it is present on 20 chromosome. You can see here it is written already. But what is this P and 13? Just write here chromosome location. Yeah. So in this diagram, you can see chromosome location. On which chromosome it is present? 7. Q is the long arm. 3 is the region. 1 is the band. After dot, there is a 2. What is that? Subband. Okay. So same, like it is written P arm. So this is what now? Short arm, because Q is the long arm. Okay. You can see Q is the long arm. Then 1 is the region. 3 is the band here. Did you got this location part? And also, if somebody asks you total exon count, so that number you can get it from here. Total 2 is the exon count. Next, what is the direction of this PRNP? Can you tell me what is the strand information, whether it is present in the forward direction or the reverse direction? Can you tell me from this diagram? In yesterday's session, we have already discussed like DNA is having two strands. If the direction it is present like this, so that means it is in the forward direction. But if it is present in this direction, it is present in the reverse direction. Now you can see in this diagram, this is your chromosome 20. Chromosome 20 start position, and this is the end position. And on this chromosome 20, there are a lot of gene that is present, RP, LOC, right? So lot of genes is present. But we are interested with the PRNP, right? So you can see this PRNP gene, it is highlighted with the blue color, and it is present here in the forward direction. You can see it is clearly written in that it is present in the forward direction, right? What if I want to get the information for this RPL? So what I will do, right click and just open in the new tab. Now this is for the RPL. And now if I want to know the direction of this particular gene, so you can see it is present in the reverse direction, this RPL, right? And also, if you get confused from here, from the diagram, you can also get the information from this table. So you can see the RS ID they are giving you. This is the assembly ID they are giving you, and the status is current and the previous assembly. So never go with this previous assembly, because this is having the older information. As you know, the research is going on. Every time new things are getting added in the genome, and the assembly is getting updated day by day. Okay, so we are never interested with the older information. Always go with the current assemblies. So in this table, you can see there are two current entries again, right? So that time you have to go with this GRCH. Why? Because this GRCH will give you complete assembly information, whereas this T to T will give you what? Telomere to telomere information. So whenever you are interested with the entire assembly, go with this GRCH. Now you can see it is present on chromosome 20. NCBI chromosomal ID they are giving you. This is the start position. This is the end position of this this RP gene on this chromosome 20. Along with that, the complement word you can see here, right? If you see this complement word here, that means that particular gene, it is present in the reverse direction. Okay. And if I go back to this PRNP, you can see there is no complement word here. Here it is written, right? So that means this PRNP, it is present in the forward direction. Did you got this? All of you? Anything you want to ask, guys? Okay. So now we will discuss how many transcript are there for the PRNP. So that time go with this transcript table. You can see total 6 transcript is there. And along with that, you are getting the gene ID, symbol name. This is the NCBI mRNA ID, that is the transcript ID, which is coding for the further protein, that is the NP, and protein name you can see here. So that means total 6 transcript is there for the PRNP. Can you give me the count of the BRCA2? Can you try for the BRCA2? Can you tell me what is the total count of the transcript of BRCA2 gene? Please try in your system. What is the count of the transcript of the BRCA2, guys? Are you there? Okay. So what if I need a faster sequence of this PRNP? That time you have to go scroll down and you will get a faster here word. So FASTA is a two-line format. First line, it always start with the greater than sign along with the ID you will get. And second line, it always start with the actual sequence of the particular gene. Okay. So now you can see this is the FASTA sequence of the PRNP. And if I want to download this, you can download it from here. Send to complete record, file, FASTA, and create file. So see, it has downloaded this PRNP. You can open and check it if you want. See, it has downloaded. So it's fine for the forward gene. Okay. What if I want to get a FASTA sequence for the BRCA1 gene? So go with the gene here and type BRCA1. Again for this BRCA1, I need the FASTA sequence. So you can see FASTA word here. Select that. And you can see this is the FASTA sequence for the BRCA1. But if you notice very carefully, this start position and the end position, it's not the same here. Can you see? It is reverse here, right? So this, it is ending with 3 to 7 here in the start position. So that coordinate, it is present in the end position. So it is totally reversed here. So that means it is not the actual sequence for the BRCA1, right? So that time, whenever you are having any reverse gene, make sure you uncheck this and update view from here. And now if you see carefully, the start position and the end position, it is matching with this table. So this is your actual sequence of the BRCA1. Remember that whenever you are using the NCBI and you are having any reverse gene, make sure you need to uncheck this and update view from here. And then you directly download from here. Complete record, file, FASTA, create file. It will download the sequence. Did you got this? All of you? Now we will see what is the largest transcript of this PRNP. So again for that you have to go to the transcript table. And this is the total 6 transcript, right? On which basis we will tell like which transcript is the largest one? Can anyone tell me on which basis we are going to tell? length of the nucleotide. Okay, you can see this is the length of the nucleotide, and 2659 is the largest one here, right? So we can assign this particular transcript is the largest transcript because it is having the largest length of nucleotide. Now simply copy this ID, go back and type here Control F and paste this ID. You can see it is highlighted. Then you can open this ID from here. Now what is the length here? You can see 2659 base pair is your length of the largest transcript. This is the largest transcript ID and this is the length of your largest transcript. Again, if I want to get the FASTA sequence of this largest transcript, go with this FASTA here. And this is the FASTA sequence of your largest transcript ID. Now you are having the length of the transcript. So in the transcript, there is a CDS region, that is a coding sequence region. So now we have to find out the start and the end position of this CDS, right? So if you scroll down, you will get a start and the end position of the CDS, that is 292 and 1053. So this is the largest transcript ID, and on this largest transcript, the CDS coordinate is this 292 and 1053. And toward this direction, there is a 5 prime, right? And towards this 5 prime, there is 5 prime UTR region, that is the untranslated region, which is not coding for the protein. And towards this direction, there is a 3 prime. This is what? 3 prime UTR region. 5 prime is your upstream and this is your 3 prime, okay, downstream. Now in this question, what we have to find out, the start and the end position of this 5 prime UTR and the start position and the end position of this 3 prime UTR. This we have to find out. Did you got this question? Now, how to get this end coordinate of this 5 prime UTR? See, if you go this side, coordinates will decrease, right? So, for this, the answer is 292 minus 1, which is equal to 291. So 291 is the coordinate of this 5 prime UTR. This is the end coordinate of this 5 prime UTR. And now for this, you have to find out the start coordinate. You are going this direction, right? The coordinates are increasing. So it is increasing from 1 till here to 659, right? So for this, the answer is 1053 plus 1, which is equal to 1054. So 1054 is the start position of this 3' UTR. And remember that wherever there is a 5' here, so from where it is getting started, so same coordinate will be there for the 5' UTR, that is the 1 here. And wherever it is ending, there will be the same coordinate, 2659 will be the coordinate for your 3' UTR end position. Did you got this part? 5' UTR and the 3' UTR, all of you? So we are left with the how many single nucleotide variants belong to this gene. In the table of content, you can see there is a variation option. Just select that. See the variants in the clean bar. Mm. So for this PRNP entries, you are getting total hits 210, that is for the germline here. So now we are interested with the single nucleotide variants, right? So that time you can see there are lot of filters here on the left hand sides, and in that you can see single nucleotide variant. So if you just select this, you will get only the single nucleotide variant mutation, you can see here. So total 160 hit is there for the single nucleotide variant. But here you can see it is written benign, uncertain significance, and somewhere you can see the pathogenic word also. So benign means that type of mutation, it is not causing any disease. So you can ignore this type of mutation whenever you get that benign word. So benign is not harmful. But if you see pathogenic word, pathogenic means that particular change of base or the mutation is causing the disease. That time you have to be very careful and you have to take those patients to the hospital. And if you see the uncertain significance, that means that particular change of base or mutation, it is still like validation, or you can see the still the proof evidence is not there proper for that particular mutation. So if you are not that much confident of that particular mutation, that is coming under this uncertain significance. So as you know, like we want to get the hit for the single nucleotide variant, that is the 160 hits. Now out of that, I want only the pathogenic. Just select that pathogenic entry. And you can see total 20 hits are only for the pathogenic that is causing the single nucleotide variant. So you can see the variation first column along with the transcript ID, the gene name, and this is the position where it is changing. Okay, 305, this is the codon position where C is getting replaced with the T here. There is a change of base, right? And then P stand for protein. Okay, the proline it is changing to leucine at the 102 position. Okay, so this is the gene name, this is the single nucleotide variant, and this type of disease they are causing in the human. And in the review status, you can see somewhere 2 star, 1 star, right? What it is denoting? 2 means multiple submitter, multiple researchers, they have submitted the data, okay, and they have no conflict regarding this particular mutation. Okay, they are very particular sure that this particular mutation is causing this disease and this is very harmful in the patient. Whereas if you see this single star, there is no assertion criteria provided, man means there is no single proof for this particular mutation. Okay, so more the star, more the confidence you will get here for the hits. Now for example, if you open this, you can see there is it is having no data for the somatic one. This is the data which has been collected from the germline database. Okay, the germline. Now transcript ID you are getting, this is the gene name. On this position, G is getting replaced with the A, and then this is the protein, it is getting changed from this to this at 180 position. This is the variant ID, single nucleotide variant, and it is present on chromosome 20. So firstly, when it was submitted in the ClinVar, you can see last submission and the last evaluated. So this type of mutation is what missense. So we have discussed yesterday also. If one sequence is coding for the proline, okay, and further second sequence is coding for the valine, so what it is causing? It is changing the protein, right? So that time that mutation is very harmful because particular base is changing what the entire protein structure, right? You can see here there is a change of protein here at 180 position. That is why it is missense here. You have to be very careful with this particular type of mutation. Then again the RSID you are getting, again from this RSID also you will get the mutation information. And if you go down, so this is the research article they have listed here in this submission. You can see in the 2016 it is a pathogenic. So never go with the older entry, always go with the updated one. So I will prefer this one, 23, because this is the latest article. So you can see it is written pathogenic, right? Along with that, every time you have to read this also information. You have to cross check with your data. If particular like protein it is changing, same in your case also, right? So that time you can assign that particular variant as the pathogenic one. Did you got this? All of you? Any doubt or anything you want to ask? Please practice this question for the other gene, maybe for the BRCA1 and the BRCA2 guys. No doubts, guys. Rajeshwari. Akhila. Okay, then. See you tomorrow, guys. If you don't have any doubt, we can leave this session. Okay, bye bye.